Hello my dear students, welcome back to my channel Biology with Chinook, and today we are going to discuss about chromatin, nucleosome and histone proteins. So let's get started. In case of eukaryotic cells, the histones and the non-histones are the two major types of proteins that are associated with DNA in chromatin. The histones and the non-histone proteins play a very important role in the process of DNA replication and help to determine the physical structure of chromosome. Chromatin is a lower order of DNA organization and it is found throughout the interphase of cell cycle. The interphase is the non-dividing phase of cell cycle, and in this phase the growth of the cell takes place and cell prepares itself for the division. So it is a very important phase and the cell enters in this phase before the division. The M phase is the phase where the cell actually divides. The chromosomes are the higher order of DNA organization. The chromosomes are formed during the cell division and appear in the prophase stage of mitosis and the leptotin stage of prophase 1 in meiosis. The chromatin is the substance that consists of DNA, proteins and RNA. The major proteins in chromatin are the histone proteins. 5 main types of histone proteins H1, H2A, H2B, H3 and H4 are associated with eukaryotic DNA. The main function of histone proteins is to help in the process of DNA packaging. How do histone proteins help in the process of DNA packaging? Histone proteins are the basic proteins that are found within the nucleus. Histones are positively charged due to the presence of amino acids lysine, arginine and histidine, and their positive charges allow them to associate with the negatively charged DNA. Why is DNA negatively charged? DNA is negatively charged because the phosphate groups in the DNA backbone carry the negatively charged oxygen. Each phosphate group contains one negatively charged oxygen atom and the entire strand of DNA is negatively charged due to the repeated phosphate groups. The backbone of DNA is made up of sugar molecules connected to each other by using phosphates. The sugar-phosphate backbone forms the structural framework of DNA and it defines the directionality of the DNA. Histones are positively charged and DNA is negatively charged. So the histone proteins bind with the DNA appropriately. The positively charged histone proteins strongly adhere to negatively charged DNA and forms complexes called nucleosomes, the basic structural units of chromatin in the eukaryotic cell. At this stage, can we see the chromatin? Yes. At this stage, we can see the chromatin with the help of electron microscope. Under the electron microscope, chromatin in its extended form looks like beads on a string structure. The nucleosome is about 11 nanometer in diameter. Under the electron microscope, the nucleosomes appear as the beads on a string structure of chromatin. Nucleosome contains a core of 8 histone proteins, 2 copies of each of H2A, H2B, H3 and H4, and this 8 protein complex is called histoneoctamer. around which a 147 base pair segment of DNA is wound about 1.65 times, a little less than the complete 2 turns of DNA around the histoneoctamer. The nucleosomes are connected with the help of linkerdna, and the linkerdna looks like the string of the beads on a string structure under the electron microscope. The length of the linkerdna varies within and among the organisms. Now, the question is, what is the role of H1 histone protein in the process of DNA packaging? H1 histone does not make up the nucleosome bead, and it is not the part of histoneoctamer, but it plays an important role in the process of DNA packaging. And we are going to talk about this in my next video. Till then, bye bye students, and thanks for watching my video.